So, before we go ahead with today's discussion, we need to refresh our memory about a few terms and concepts. So, the first major term is that of the gene. Now, nowadays when anybody thinks about the gene, what they think about is a stretch of DNA which is transcribed and either codes for a RNA or codes for some kind of a polypeptide which either has a known function or a presumed function. Now, this is a very mechanistic way of looking at a gene, and although this is what is mostly molecular biologists, this is what they prefer. This is not how evolutionary biologists typically think of a gene, or for that matter even the transmission genetics people, they do not think about gene this way. The way they think about it is what we call a Mendelian gene, which is a genetic unit which is transmitted from parents to offspring and which is detected through phenotypic differences associated with different alleles at the same locus. In other words, if you have multiple forms of that gene, then they are defined in terms of having their effects on the phenotype, not simply having an differences in the sequence. Now, in this particular course, we are going to use the term gene both as a unit of inheritance and as a mechanistic unit of some kind of a transcription leading to a protein or a RNA. So, how will you know in which way I am using it? So, whenever I say gene in general, I will be talking about a Mendelian gene. But suppose I tell you specific gene's name, okay, for example, the serotonin transporter gene that we discussed in the last discussion. When I give a specific name, that is when we are talking about a molecular gene. But when I say gene in general, it is the Mendelian gene in the context of a transmission unit. That is what we are going to talk about. So, this definition has two more terms, alleles and locus. So, what are they? So, allele is simply one of the several alternative forms of a gene that is present in a population. So, for example, again last discussion we talked about two alternative forms of the serotonin transporter gene. One was the L form and the other was the S form. So, these two are two alleles. And then what is the locus? So, you know that the genes, they are arranged on the chromosomes and they have specific positions on the chromosomes, and these specific positions of a gene or a genetic marker is what is known as a locus. Two other important terms, one is genotype. What is a genotype? Simply the genetic makeup of the organism for a particular locus or multiple loci. So, for example, in the context of Mendel, remember he said that he started with true breeding tall plants and true breeding short plants, and he said that the true breeding tall plant has capital T allele at both the both positions on the homologous chromosome. So, at the same locus it has two copies of the capital T allele. So, this thing, this capital T or a small t is what indicates the genetic makeup of the organism at that particular locus, and hence it is the genotype of the pea plant. Now, what is the phenotype? The phenotype is simply an observable trait or characteristic of an individual or an organism. So, for example, in the context of height, in the Mendel example, tallness is a phenotype, shortness is a phenotype, right. Now, if you remember, we said that the rediscovery of Mendel's laws were exceedingly important for evolutionary biology. So, before we go ahead, we need to quickly have a look at what exactly Mendel had said. So, if you look at Mendel's three laws, the first law is the law of dominance, which says that in a heterozygote, the phenotypic effect of the recessive allele is masked. In other words, if you have, let us say, the capital T plants being tall, the small t plants being short, then when you made the capital T plants with the small t plants, then in the next generation, the offspring generation, the F1 generation, the genetic makeup of all the individuals will be capital T small t. So, they are going to be like this, the heterozygous form, yet phenotypically all of them are going to be tall. In other words, the effect of the small t allele will be completely masked by the capital T allele. Now, remember, this is what is happening at the time of Mendel. Today we know that this is not always the case. So, many a times when you have a scenario of a heterozygote like this, the phenotype of the heterozygote is actually intermediate. So, nowadays the specific thing that Mendel saw, we call it complete dominance, when the dominant form is able to completely mask the phenotype of the recessive form. However, nowadays we know that you have different degrees of incomplete dominance, and in all those cases the phenotype of the heterozygote is actually some kind of an intermediate between the phenotype of the dominant homozygote and the recessive homozygote. So, this is the law of dominance. Now, you have the law of segregation. So, suppose we are talking about one locus scenario, a diploid organism. So, the organism has 2 copies of that particular gene. Now, what Mendel says is that during the process of gametogenesis, that is when the sperm cells or the ova cells are formed, then the cells have to undergo what is known as a reduction division, right. So, each gamete actually has only 50% of the genes that the original organism had, and in the process of the formation of the gamete, each gamete will get only 1 copy of the gene, whereas the parent would have had 2 copies, right. So, this is the law of segregation. And finally, suppose you are talking about multiple loci, then you have genes at these loci, alleles at these loci, and what Mendel said was that during the process of gametogenesis, the alleles at different loci are going to assort independently, which means that they are going to go or divide themselves into the gametes irrespective of, independent of what allele is present at the other locus. So, the allele at any one locus is going to get into the gametes without any effect of what allele is present at the other loci. Now, today we know that this is true only when the loci are on different chromosomes, or the loci are so far away from each other on the chromosome, let us say on 2 ends of the chromosome, that a crossing over event is highly likely. If that does not happen, if the loci are on the same chromosome and are relatively close to each other, we get a different phenomena called linkage. However, Mendel did not talk about it. Mendel explicitly looked at those loci which were either on different chromosomes or were very far from each other. So, this is where our revision ends, and now the discussion for this particular topic begins. Now, if you look at all this that I talked about, Mendel's work, this is in the context of transmission of genes within a family. Why am I saying this? Remember, what Mendel was doing was, he was taking true breeding varieties, and then he was taking other true breeding varieties, he was crossing them, and he was trying to see what is happening at the next generation. And then he would self cross them, and he would look at what will be happening in the next generation. So, in all the cases, he is having full control over who is mating with whom, and he is simply looking at what the offspring are looking like, right. However, if you think about a population, a real biological population, we do not really know, or we do not really have any control over who is mating with whom, right. So, if you are thinking about looking at evolution from the perspective of changing genetic variation, remember that is what I said, you can think about evolution as how variation is changing across generations. So, if you are trying to do that, then conceptually it is not very difficult. Conceptually, what do you have to do? Conceptually, you have to go out there and measure every single individual and look at what their genotypes are, and then you have to look at all their babies, and in the next generation look at what their genotypes are, and you can simply compute what is the change in gene frequencies. Now, of course, empirically this is going to be very expensive, this is going to be very troublesome, but conceptually there is not much of an issue. However, when you try to think what is happening under the hood, then you realize that this is, biologically speaking, this is not a straightforward process as it looks like. Why is that so? Because underlying what is happening is something like this. You have the male, I am talking about sexually reproducing diploid organisms. So, you have the male individuals, you have the female individuals. The male individuals, they are undergoing meiosis. So, during the process of meiosis, two things are happening. A, crossing over is happening, because of which the chromosomes are all getting jumbled up, the homologous chromosomes are all getting jumbled up, and then B, reduction division is happening, because of which each gamete is getting only 50% of the genome that the original organism had, the parent organism had, and that stuff that it is getting, that genetic composition that it is getting, is actually all chopped up thanks to crossing over. This is happening in the males, this is also happening in the females, right. So, now you have lots of gametes, but then the organisms are mating, and they are mating in with whosoever they please, and even when they are mating, not all the gametes are forming the zygote, only a subset of the gametes are forming the zygotes, and then these subset of the zygote, a small fraction of that is actually taking birth, and then they are growing up to form the next generation, and you sum over all the mating pairs, and only then you get the next generation, right. So, biologically speaking, this process is a lot more complicated and therefore very difficult to track, okay, if you are going to track family by family. So, what you need to do is to come up with a way by which this entire process that is happening over here at the level of the family, this entire process can be somehow abstracted out such that you can go directly at the population level from generation T to T plus 1. And the branch of evolutionary biology which does that, which takes you from generation T to generation T plus 1 at the population level, somehow taking care of all the nitty-gritty details, gory details of the biology at the lower level, that branch of evolution is what is known as population genetics. Simply speaking, it is extending the concepts that you get from Mendel's laws from the family all the way to the population. Now, how does one do that? How does the subject do that? Now, in order to do that, what it does is certain abstractions, okay, and these are very, very powerful abstractions. Conceptually, they look trivial, but once you see the kind of insights they can give you, you realize that it's absolute marvel. So, what is the nature of the abstraction? So, first we will talk about the nature of the abstraction, and then in the subsequent discussions we are going to see what comes out of it. So, what is the nature of the abstraction? So, suppose we are talking about a one locus scenario, right. Now, if you are talking about the organism, an organism obviously does not have just one locus, it has many loci, many genes all interacting with each other in a very complicated way. But population genetics says that when you are thinking about a one locus scenario, then just think about that one locus, forget about everything else, right. Now, if you have a diploid sexually reproducing organism, then in that population, if you are thinking each individual and you are concentrating only on that locus, so you know that for each locus you will have two homologous copies of the gene, right. So, you think of each individual as some kind of a pair like this, right. And here let us say we have two alleles, we can have more, it does not really matter, but for the sake of discussion let us assume that we have two alleles. I am representing one as shaded, the other as white. So, these are the various genotypes that we are going to get. Of course, I am showing you just 3 individuals, but there can be many more such individuals. Okay. So, what is the implication of this? So, if you think of the organism like this, so this is the parents, let us say, then what will the gametes look like? Remember, the gametes are simply happening or coming into being because of reduction division during meiosis. So, according to Mendel's laws, the gametes are just these genes separating out from each other and some of them in this particular case will be dark because we have 2 alleles, right. So, the gametes are simply the genes, these genes. Now, you have this happening for the males and this happening for the females, right. So, you have all the gametes produced by the males, you have all the gametes produced by the females. Okay. Some of these will be dark like this. Now, at the level of the biology, mating is happening, okay. But at the level of this particular locus, how do you conceptualize mating? At the level of this particular locus, mating is simply taking one gamete from the male pool, which is this one, taking one gamete from the male pool and uniting it with one gamete from the female pool, right. So, when you are looking at the next generation, then essentially all you have to do is to pick up one gamete from here and one gamete from here and join them to form the next generation, right, something like this. It all goes back. So, this is the offspring generation. Now, why should I think about it like this? What benefit do I get? The benefit that I get is that if you think about the whole process like this, then mathematically all these things that going from parents to the gametes, going from male gamete, female gamete to the next generation's offspring, all these essentially become processes in sampling. Okay, you are simply sampling some gametes from the overall pool of the gametes. You are simply sampling some male gametes and female gametes and putting them together. And the moment you get into the domain of sampling, then we have the entire mathematical tools of the science of probability of available to us. And because we know a lot about how probabilities work, we can actually then deal with all these things using the maths of probability. So, this is precisely what population genetics does. It takes a problem which is in the domain of biology, and by making these assumptions, making these mental constructs, it actually translates them into stuffs in the domain of probability. And of course, it is much easier to handle them in the domain of probability. So, the entire process of transmission of genes over a large number of mating pairs in the population, that just gets abstracted down into sampling from bag full of gametes. Now, at some level, many people find this somewhat discomforting, because biology is complicated, so many genes, behavior, everything is determining who is mating with whom, behavior, culture, etc. And yet at the level of the locus, you are just putting all those complications under the carpet and you are just focusing on sampling. And that is why some people very derisively call population genetics as beanbag genetics. Okay, it is simply sampling beans from a beanbag. But wait till you see how powerful this conceptualization is, and the kind of things, the kind of results that it leads to, which can then be tested both empirically in the lab and empirically in the field. So, a lot of what we are going to do over the next 2-3 weeks is going to be in this framework, trying to first derive what is the prediction, and then explicitly checking it out using simulations and explicitly looking at real life data to see whether that prediction holds or not. But before we get there, there is a very simple result that we need to derive. It is a very simple result, but again it is a very powerful result and an important result. So, let us assume that we have a 1 locus 2 allele scenario, and the locus is autosomal, it is a sexually reproducing organism, and the generations are discrete, which means that parents and offspring never mix. Now, let us further assume that the 2 alleles that we have in hand, they are called A1 and A2 respectively, and the frequency of A1 and A2 are small p and small q. Let us then understand that if you have 2 alleles A1 and A2, then you are going to have 3 genotypes A1A1, A1A2 and A2A2, and let us assume that the frequencies of these genotypes, the proportions of these genotypes in the population are represented by capital P, capital Q and capital R, as I am showing you. Great. So, if that be the case, then certain things are very obvious. What is obvious? We have just 3 genotypes in the population, right. So, the sum of the frequencies of 3 genotypes, that is P plus Q plus R, has to be equal to 1. There is nothing else that can be, right. Similarly, if you have 2 alleles A1 and A2, then the frequencies of these alleles have to sum to 1, P plus Q is equal to 1. So, this is kind of obvious. What is a lot less obvious is how we are getting to this result, that small p is equal to capital plus P plus Q by 2 and small q equal to capital R plus capital Q plus by 2. Now, there are lots of people who think in terms of ratios and proportions and simple algebra. They will take one look at this relationship and say, yeah, it is very intuitive. However, there are many more people, and I am one of those, for whom it is not really obvious at one glance how small p is equal to capital P plus Q by 2. So, what I will do is I will derive it very easily for you, and that hopefully will make things very clear. So, let us assume that the number of individuals of the A1 type in the population is N11, the number of individuals of the A1 type is N12 and the number of individuals of the A2 type is N22, right. So, if that be the case, then the total number of individuals, we will call it as N, is equal to sum of these 3, right. So, N11 plus N12 plus N22, right. Now, we want to figure out what is this frequency of the A1 gene. We want to figure out what is small p. Now, this small p is equal to total number of A1 alleles divided by the total number of alleles, right. Now, what is the total number of A1 alleles? Remember that the A2 folks, they do not have any A1 allele, right. So, we can disregard them. The A1 folks and the A1 folks, they are the only ones that have the A1 allele, but they have it in different numbers. So, each A1 individual has 2 copies of the A1 allele, right, and we have N11 number of A1 individuals. Therefore, the contribution of the A1 homozygote is 2 into N11, right, and what is the contribution of the A1 heterozygotes? Each one of them is giving me 1 copy of the A1 allele and there are N12 number of individuals. So, the total that we are getting over here is 2 into N11 plus N12. What is the total number of alleles? Remember, we are talking about diploid organisms. So, each one of them have 2 copies of the alleles, of any allele, and therefore, the total number of alleles is 2n. Now, this thing I can write it as 2n11 divided by 2n plus n12 by 2n, correct. So, this 2 and this 2 get cancelled out. So, this becomes n11 by n plus n12 by 2n. Now, here what is n11 by n? N11 is how many A1 individuals are there and n is the total number of individuals. So, this n11 by n represents the frequency of the A1 individuals in the population. In other words, this term is equal to capital P. Similarly, here if I just look at this part n12 by n, there are n12 number of A1 individuals and n is the total number. So, this is the frequency of the heterozygotes n12 by n and this is q and have this extra 2. So, this is P plus Q by 2, right, which is what we derived over here. It's very simple once you think about it, but this is actually a very important result, and as we will see in the next discussion and the one after that, many a times students end up ignoring this particular result, and therefore they end up getting completely wrong answers to certain kinds of questions. We are going to deal with those questions explicitly, but what I want you to do is to satisfy yourself just using the same logic that we used over here, that small q indeed is equal to capital R plus capital Q by 2. Okay. So, with this result, we are now in a position to extend Mendel's laws to the entire population level, but that is what we are going to do in our next discussion. See you there. Bye.